So in this fourth part of chapter 11, we're going to now take a look at the steps involved in meiosis 1 and 2. These are figures from your book. Feel free to look on the internet for additional resources. Your lab book even has some really good explanations of this. So following interphase, a cell will enter meiosis 1. And remember, we're starting with a diploid cell, so the replication of the genetic material has already occurred, and our replicated chromosomes are beginning to form as the nuclear envelope erodes during prophase 1, very similar to what we saw in mitosis. The main difference is that during prophase 1 of meiosis, as our paired homologous chromosomes are beginning to form, they align with each other, forming something called a tetrad. And the tetrads actually allow for the sister chromatids from two different portions of the homologous pair to do something called crossover. Now, this crossing over that occurs between these non-sister chromatids, and we'll take a closer look at this in just a moment. But basically what's going to happen is this, at this point of crossing over called the chiasma, these non-sister chromatids will actually swap pieces, making some brand new combinations. Right? So this is one of the main differences between prophase 1 of meiosis and prophase of mitosis. Now, following prophase 1 is metaphase 1. And very similar to what we saw in mitosis, we're going to see the chromosomes align themselves at the equator or the metaphase plate of the cell and attach themselves by the centromeres to the microtubules or the spindle fibers. Except now, instead of the individual chromosomes aligning, it's the tetrad that's aligning. And recall that this word tetrad refers to tetra, which is 4. What do we have 4 of? Well, we have 4 sister chromatids here in our tetrad. Now, following metaphase 1 is anaphase 1, and in anaphase 1, it's the homologous chromosomes that are going to be separated as the microtubules of the spindle fibers pull them towards opposite poles of the cell. We see now that the sister chromatids that are being pulled in these different directions actually have the pieces of the other chromosome attached to them, denoted by these different colors here. We also see the cleavage furrow beginning to form, so this is obviously an animal cell. And following anaphase 1, we see telophase 1, where the nuclear envelope will begin to reform and our chromosomes will relax a little bit. We'll see cytokinesis occurring, separating the two cells in the one cell into the two cells. Now, following interkinesis, we now enter meiosis 2. During meiosis 2, we begin with prophase 2, and there's two cells being shown here to remind you that we started with one cell. At the end of meiosis 1, we have 2 cells, and at the end of this, meiosis 2, we're going to have 4, right? So keep track of that. Now, during prophase 1, our chromosomes are all aligned here. The nuclear membrane breaks down, and the spindle fibers begin to form. And during metaphase 2, our chromosomes are attached to the spindle fibers. Just like in metaphase 1, there is a random assortment of the direction at which the sister chromatids face. Notice here, this blue maternal chromosome and this with a piece of the paternal chromosome here could be pointing in different directions. This is something that we'll talk about in just a moment called independent assortment. Again, it's going to produce some genetic variation. Now, following metaphase 2, the sister chromatids will actually be pulled apart during anaphase 2, and during telophase 2, we see the haploid nuclei begin to form as cytokinesis is completed. Now, again, there's two very important processes that occur during meiosis that promote this genetic variation. And I mentioned them when we were looking at those figures, but here we list them. Crossing over, which occurs during prophase 1, and the independent assortment of the chromosomes that we see during metaphase 1 and 2. So let's look closer at crossing over now. So crossing over occurs during prophase 1, and this is where those replicated homologous chromosome pairs line up and are physically connected. This process to form the tetrads is known as synapsis. So as these homologous chromosome pairs synapse to form tetrads, you can actually see the non-sister chromatids being attached at this point called the chiasma. The chiasma is the point where the non-sister chromatids overlap, and we see this genetic rearrangement occurring. Literally, the maternal and paternal chromosome will swap pieces. Now, what this does is it produces something called a recombinant chromosome. Chromosomes that now carry genes from two different individuals, or in this case, two different parents. So, our tetrad then consists of 4 chromatids, 2 pairs of sister chromatids. The site of crossing over is the chiasma, and during synapsis, these recombinant chromosomes are going to be formed as they swap pieces. Now, during anaphase 1, it's actually these homologous chromosomes that are going to be pulled in different directions. Now, what about independent assortment? Well, independent assortment refers to the direction at which these chromosomes line up on the metaphase plate. And again, it's just north or south, heads or tails. There's a 50-50 chance whether each one of these homologous chromosome pairs, or in the case of what occurs during metaphase 1, the tetrads themselves will begin to move. So the chromosomes all sort independently. And if you think about it, we have 23 pair of chromosomes, homologous chromosomes in a human. So each one of those pairs will sort independently of one another during metaphase 1 as tetrads and during metaphase 2 as sister chromatids. Now, all of this promotes genetic diversity. And what this means is that our daughter cells then have all of these unique combinations of different genes. And when you couple this with the crossing over that's occurred, it really helps us to understand how genetic diversity is promoted here. Now, if we look at the possible number of combinations when chromosomes sort independently, it really helps us to see how truly unique we are. You can use some simple math here. 2 to the nth power gives us some of these probabilities to look at how likely it would be for combinations to occur by random chance alone. So in humans, where N is the haploid number of the organism. In humans, we have 23, our haploid number is 23. We have 2 to the 23rd power, which is about 8,388,000 different combinations that could be produced. So if you couple this with the probabilities that factor in crossing over and then random fertilization, 1 sperm from a male and 1 from a female, you multiply those together, you can see that it's literally greater than a 1 in a 70 trillion chance that 2 of the same parents could produce an offspring with exactly the same genetic combination. It reminds us how unique we are, truly. Now, this last slide here is just a good slide for you to review that summarizes all of this stuff. It also compares and contrasts mitosis and meiosis. Recall that crossing over using these tetrads occurs during meiosis. And during anaphase 1, this is where we see the homologous chromosomes separate. Remember also that in meiosis, we have a reductional division, taking the cell from haploid to diploid, that occurs in meiosis 1, followed by an equational division in meiosis 2, where those haploid cells split. So make sure you review these and compare and contrast the steps that occur in mitosis versus meiosis.